Okay, welcome lower sixth. This is me going through the paper that you sat before half term, so October 2014, and just the biochemistry side of things. It'd be great for you to watch it, have your test open in front of you, add corrections, obviously pause it whenever you can. I've just been told off for sounding too monotone, but I think it's my way of getting through it quite quickly. So there is the beautiful membrane, and name the molecule labelled A. I think that should be straightforward. That's a phospholipid. The structure, and describe its structure. So there's 3 marks there. That's the first mark. So actually it's sort of simplified with that. There is actually a glycerol within it. So you've got the phosphate head, the glycerol, and the 2 fatty acid chains. So already I've sort of got 4 marks there, and it's held together with 2 ester bonds, so each fatty acid tails. And you remember that's again formed from a condensation reaction, water being lost. Explain how the properties of the molecule A contribute to the structure of the cell membrane. Now, what you've got with a membrane, you've got an aqueous or watery environment in the tissue fluid, meaning outside the cell, and then you've got the watery or aqueous environment in the cytoplasm. And what happens is these phospholipids orientate themselves. So you have got the phosphate heads, which are hydrophilic. Okay, well, there you go. Hydrophilic or polar phosphate heads aligned so they're in contact with the water. So you're going to have the 2 sides like that. The hydrophobic or non-polar fatty acid tails orientate themselves away from the water, so inside. Okay? When it comes to proteins in the cell membrane being involved with active transport and facilitated diffusion, active transport, so I've now simplified it, just on 2 lines, but of course you've got all the phospholipids in there. What you have is a protein carrier or pump which takes molecules from a low to high. So this is against the concentration gradient, that's the key term there, and it uses energy. So you imagine you need to pump that in to get it against the concentration gradient, and there's ATP in there. Another detail you can say is that it's specific, these solutes or whatever they are, they fit into the protein carrier. So that is a very specific pump. Facilitated diffusion, what you've got is the idea of protein channels, or you've got a protein carrier as well, but the big difference, it's down the concentration gradient. Diffusion is high to low, okay? Active transport low to high, really clear. And I think that was a bit triple award biology at GCSE, and so people who didn't do triple award, you really need to engage with this to catch up. Okay, and these molecules are large polar molecules when it comes to that. So this is a little experiment that's been done. You've got 2 cells, cell A and cell B, and they've stained the proteins. It's very classic, it's staining things. We often put a fluorescent tag on things. And so basically what they do is they're pushing them together. Ooh, and look what happens. So you've got the uneven distribution, or what's happened is that the red and the green have intermingled. Why does the result of the experiment provide evidence of the fluid mosaic model? And obviously that is that concept that all the proteins move around within the membrane, within those phospholipids. Why? It's the phospholipids which allow the fluidity, because they're not actually bonded together. If you tap them, they will move apart. There's a great YouTube clip which I'll put on Firefly, which is the new learning platform that will have lots of resources to help you with revision. So they allow the fluidity which allows a membrane to fuse. Okay? So if they were strictly bonded between it, if you try to push them together, they wouldn't fuse like that, would they? They just stay as 2 cells separate. But because there's fluidity, because if you tap it they fall apart, then you know that would happen. So fluidity allows the proteins to move and intermingle. Okay? There is your lovely phospholipid bilayer again, or the fluid mosaic model with it all in there. The fluidity of the membrane is determined by the proportion of cholesterol. So the phospholipids allow it to be fluid, but having cholesterol changes the fluidity. You can see sort of they're interacting with the phospholipids, I always think of it, like a membrane that wobbles. And if you've got, this depends on the temperature, but certainly different amounts of cholesterol determine on whether it's sort of less fluid or more fluid. Okay? So then you've got this experiment, that this is the core practical that you all did. And it's essentially you've got a student 1 and student 2, carrying out the practical differently. Of course, every practical can be. And it's that sense of you now applying your how science works knowledge, which is going to be developing throughout this 2-year course. Use the information, describe the effect. Now, describe means, really just don't apply any biology, literally just say what it is. And there you go. An increase in temperature increases permeability, because they're asking for the effect on permeability. From 42 to 93. Okay, so that sense of the colour of the fluid surrounding only gets darker from those values. It would be wrong to describe it saying from 5 to 93 the permeability increases, because from there to there there's no change. Okay? Now, of course, this is more similar to what we actually did using the colorimeter. And you've got lovely repeats there. Immediately I'd be thinking, oh, that looks better than that other experiment. State 2 variables that both of these students must keep the same, i.e. what are the controlled variables? Their rinsing method. So we all remember you boiled out the beetroot and you rinsed it. Okay, so you've got to rinse it perhaps for a minute. That would mean that the method was the same. The size of the beetroot, or the surface area or shape of the beetroot. When you're doing that, you realise that that was a way to make it possible. Same species of beetroot, and the same incubation time, i.e. the time it was in the temperature. Like if it was in the 64 degrees Celsius water bath, it'll be the same time in there, like 10 minutes. And then the pH as well. Okay, why is second student better, more reliable? So reliable, think repeats. So he had repeats or she had repeats. And it's not based on just judgment of colour. It's not subjective in the sense that it's just literally saying that, oh look, it's gone dark pink or pink. Perhaps that's just a subjective. Perhaps it was actually the same. Here we've actually got quantities, so values. And you can really base a more reliable judgment on it. It's quantitative rather than qualitative. You're not looking at the qualities, you're looking at the quantity, the value. Okay. So then this question here, with regards to the first student, it was pale pink. We mentioned that earlier. Why was it pale pink? Perhaps the first student didn't wash off the dye, which was released by cutting the cells. Okay, so when we did that washing process, you were reasoning with why that was an important step. Okay. Each of the students used their own results to describe the effect of temperature on the permeability of the membranes of the beetroot cells. Suggest one way in which these two descriptions might differ. So second student saying that the permeability increased from 5 degrees Celsius, because you saw that change in the value, whilst first student increases from 42. Okay. Now I know you groaned when you saw this graph. So solubility in oil compared to water. Describe what relationship, if any, there is between the permeability of the cell membrane and the size of the molecules. Now the bigger the circle, they've written it up here, the bigger the molecule. Now I'm looking along there, so permeability of the cell membrane is up there. Okay, is it simple that like smaller ones are down here with low permeability and larger ones are up here with higher permeability? No, look, there's some small ones up here as well, and there's some larger ones down there. There's absolutely no relationship. Okay, so be brave, think that that is a possible answer. Next question, describe what relationship, if any, there is between permeability of the cell membrane and the solubility of the molecules in oil compared to water. Now this is when you're just looking along solubility of oil. As that increases, does that increase? Yes. You can see that there is a correlation, a positive correlation. Okay, it's not directly proportional. If it was directly proportional, when it was 1, that would be 1. Okay, but there is a positive, quite a strong positive correlation there. Next question is about osmosis. And what I really like this image. It's really helpful because you can see the partially permeable membrane. these big brown blobs represent solutes like glucose, sucrose, amino acids, things that dissolve in water. And things that dissolve in water are polar. Okay, so describe, using the method describe and explain. Describe, water will move by osmosis from left to right. Okay, from a higher water concentration to the lower water concentration. Can you see how much higher water concentration, those little things, there you go, to the lower. Okay, explain why. Water is dipolar. Okay, it sticks to the polar solutes such as glucose. These solutes can't fit through the partially permeable membrane. So that sentiment that, look, you can see them all stuck on there, that basically is the thing that lowered the water concentration on this side. Okay, if you got rid of those solutes, then it'll go, or got even rid of one, you'd have just as many free water molecules. I really hope that this has helped. Let me know.